Hello and welcome to this session on kidney function, specifically in relation to excretion. It's question 4 on the alternative assessment, however we can still use it to learn about how the kidney works, how it's structured and how its structure affects its function, which is a real classic in human physiology. Put this first slide in to sort of show you a rough guide as to where they are in terms of the body. You can see that they're either side, and they're connected to this bottom thing here, which is the bladder. And the function of the kidneys really is to filter all of the bad stuff out of our blood. And all of that filtrate, it's called, gets passed down into the bladder. That's the stuff that we want to get rid of. But what actually happens is the kidneys don't really differentiate between what's bad and what's not bad. So basically the kidneys will filter everything and then pass back the good stuff back into the blood and only get rid of what it needs to get rid of. So they're really, really important for our bodies. If we didn't have them, then we'd obviously, we would die essentially. And that's why people who have malfunctioning kidneys or don't have kidneys at all have to go through the process of dialysis, which is a really sort of invasive and life-affecting condition and situation to be in. So think yourself lucky for those people that do have them and that they're working really well. So here's the criteria. With reference to the excretory system, explain the following terms. So we're going to look at those 3 terms specifically and independently. And it's the old classic: how does the structure enable it to function? What does the kidney have to do this, this and this? Okay, we'll start with a layman's terms approach, and believe me, before I sort of looked at editing this slide, I didn't realise how funny this picture was. And I'll just leave it at that. But here we've got the blood rushing down a vessel, let's just say, and what actually happens is the kidney filters the blood, and all of the small stuff in the blood will go through this sieve and into what's called the nephron. So all the small bits will go into the kidney. All the big bits that can't fit through the sieve will stay in the blood. Okay, that's the first thing that happens. We separate the small stuff from the big stuff. And you can see some examples here at the bottom: water, salts, bicarbonate, hydrogen, etc., etc. Some examples of the big stuff might be drugs, might be big proteins, things like that, hormones that are too big to fit through the gaps, and therefore they stay in the blood. Well, the second thing that happens is that some of this stuff here is really important, particularly glucose and some amino acids and water. So the body takes back what it needs. So after having filtered the small stuff, it will then pass it back into the bloodstream. And finally, there's still big stuff in this blood that we couldn't filter out, and we put that back in to the nephron, into the kidney, which then will form urine, and that's what we will excrete. So it's not just a case of really separating the bad stuff from the good stuff. It's not that easy. First of all, we filter the big stuff from the small stuff. Secondly, we put back into the blood what we want to keep. And thirdly, we put back into the kidney what we want to get rid of, and that's what is excreted. That's the sort of general principle of the kidney and how it works. If you keep that in mind, the rest will sort of become easier, I feel. So these are the 3 processes that we need to talk about. Sometimes they're talked about in different terms. So ultrafiltration is sometimes called glomerular filtration. Tubular reabsorption is sometimes called selective reabsorption, and tubular secretion is sometimes called excretion. Tubular secretion is sometimes called excretion. So just so that you're aware, we can talk about those terms interchangeably. Some interesting facts about the kidneys and how they work. They can process up to about 180 litres of your blood fluid every day, which is obviously why dialysis is really important for those people that don't have kidneys or that have malfunctioning kidneys. You know, if you think about 180 litres a day, that's a heck of a machine. And that's so that the toxins and the stuff that we don't want in the blood and in the body doesn't build up. We have to continually filter it out. Now, if you're filtering 180 litres per day and only 1% leaves the body, you can see that that's a really minute amount. But that's what it does. It sort of filters, it processes a lot of blood, but only takes out the stuff that it doesn't need. And it filters the entire plasma volume 60 times each day. So during the day, the whole volume of your plasma is being filtered 60 times per day, which is really remarkable. And as such, they require a lot of energy. There's a lot of processes going on in there. So let's take a look at how it works. So here's the macro view of a kidney. There are some distinct parts before we move on to really looking at it in fine detail. This is called this is the artery that supplies the kidney, and this is the vein that, let's do it in blue, this is the vein that takes the blood back. So the renal, just doing it in a different colour to confuse you, the renal artery and the renal vein. And you can see it's supplying oxygen. It's red and blue, so this renal artery is going away from the heart. If you've watched my video on the third lesson, A for away, arteries, it's taking oxygenated blood to the kidney. That oxygen in the blood is being used, and it's carrying deoxygenated blood, and that'll go back to the heart. But I want you to focus in really closely on how this artery branches off into different arterioles and then capillaries. So all these branches here are capillaries that are really, really important for filtering the blood, because not only is it supplying oxygen to the kidney for it to work, the blood that's actually going in is being filtered. So the blood that's coming out of this renal vein has been filtered of all its toxins. What happens? All the toxins and all of the bad stuff is going to go down this pipe here, which is called the ureter. Not the urethra, that's what you did in reproduction, and that's the sort of exit pipe. The ureter is the pipe that links the kidneys to the bladder. This is a sort of waste pipe. So in this kidney, the magic happens where this oxygenated blood comes in, it gets filtered, and the waste products go down the ureter. Okay. So we generally have 3 distinct sections to the kidney. This section here is generally called the pelvis, the renal pelvis. The next sort of layer would be this layer here, where you can see all the sort of buds picking off. This section here would be the medulla, the renal medulla, and this section around the outside, the external section, would be the cortex. The cortex. So that's a sort of large view, step back view of the kidney. Cortex around the outside, the medulla towards the middle, the pelvis really in the middle, I suppose, where the joints of the arteries, veins and ureter comes in and out. And what we're going to be talking about, the actual functional unit of the kidney, the thing that's going to be doing the filtration, is called a nephron. A nephron. And the next few slides are going to be all about the nephron. And the nephrons, and there are millions of them if I'm not mistaken, will start in the cortex here, and I'm going to draw a circle and a cup shape. And the nephron starts in the cortex, it drops down into the medulla, comes back out into the cortex, and from there it drops down into the ureter and down the waste pipe. So the nephron, if you want to come back to this slide just to sort of give you a perspective of where we are in terms of the kidney, starts in the cortex, drops into the medulla, comes back out into the cortex, and then from there goes down the collecting duct into the ureter. That's where we're talking about if you ever get lost in terms of where we are in a kidney. Okay. So I'm going to attempt to sort of draw the nephron for you to help you to be able to firstly identify the 6 or 7 key areas, key sections of a nephron that you're expected to know. So let's start with the blood supply. The blood supply goes from the renal artery, and it gets smaller. It gets smaller and smaller into capillaries, and here we've got a capillary that goes into, or is linked with the nephron. This capillary then goes into a sort of ball and then exits out like that. So here we've got, so this is called the afferent capillary, and this is called the efferent capillary. So this one's going away, this one's going into, and this sort of bundle of capillary, this ball of capillary here that you've got in the middle, is something called the glomerulus. Glomerulus. And that's what it is. That's a bundle of capillaries. That bundle of capillaries are surrounded by a capsule. Now we're only drawing it 3D, it looks like a U shape, but you can actually think of it like a hand cupping a tennis ball when you throw in it. It surrounds the glomerulus. And this capsule is called the Bowman's capsule. Now these things have a very specific job, and I'm going to spend the next few slides doing it. All I want you to be able to do is identify what's going on. We've got the afferent arteriole, I should say, the afferent arteriole leading blood into the glomerulus, and the blood leading out of the glomerulus is the efferent arteriole. Now, excuse me, what we've got around there is something called the Bowman's capsule, and actually what happens is blood gets filtered into this Bowman's capsule. And the Bowman's capsule really is the first part of the nephron. What I've talked about here in terms of the blood is not really the nephron, but you do need to know about it. That's the blood supply. The Bowman's capsule is the first part of the nephron that you really should be able to identify. Now, this Bowman's capsule then turns into a bit of a tube. Okay, and this tube eventually drops down a big U shape like that, like a roller coaster. Okay, now remember where I said we were in the kidney? This section here, let's say, I've probably done it a little bit low, but this section above this line is the cortex, the outside of the kidney. This section is the medulla. You might want to go back to the last slide just to check where we are in terms of the landmarks, in terms of the kidney, because it's really important later on when we're talking about it. So the nephron drops down into the medulla, comes back out up to the cortex, and it turns into another squiggly little tubule. And as I said earlier, this then turns in, or this connects to a collecting duct. Which is connected to the ureter, and that's where all the waste products go. Okay. The only things I haven't given names to are the 3 sections of tube. I just call them tubey things. Here, 1, 2, and 3. This tube here is called the PCT, the proximal convoluted tubule. I'm afraid I'm drawing with a mouse, so it's not very accurate. Convoluted, and that's a D, would you believe? Tubule, proximal convoluted tubule. Proximal, you should remember from skeleton and muscles. Proximal means near, and it's near to the Bowman's capsule. So the little tube coming off the Bowman's capsule is definitely the proximal one. Sometimes you'll see diagrams that are the opposite way around, the mirrored way around. So sometimes you'll see the Bowman's capsule on this side of the diagram, and you'll see a tubule coming out this way, maybe. I've not done that very well, like that way, and that would be the PCT, you know, and the diagram will be going that way, just so that you know if you see it in textbooks or anything like that. This big drop here, this roller coaster, is called the loop of Henle. The loop of Henle. Specifically, this is the descending loop, the one that's going down. The one that's going up is called the ascending loop of Henle. Okay, and finally, this one up here is called the distal convoluted tubule. Again, distal in biology means furthest away. I'm not going to write it because I made a right mess of the other one. But distal convoluted tubule, this is this section here along the tubule. So really you've got, what, 5 sections of the nephron that you need to know about. Number 1, the Bowman's capsule. Number 2, the proximal convoluted tubule, or the PCT. 3, the loop of Henle. 4, the distal convoluted tubule, and 5 would be the collecting duct. The only other thing that I would expect you, or ask you to know about, it would be the glomerulus, so 6, and the fact that the afferent and efferent arterioles go into and out of that. All right, so there we've got it. We've got a nephron. 7 things, maybe 8 things to remember if you include the ureter. And that's sort of how the locations of the nephron. Okay, so the first thing that the question is asking you to explain is the process of ultrafiltration. So what we're going to do is explain that process and look about how the body is suited to do that process. So here on the right you have the nephron, just as I've sort of explained it. It looks a little bit neater than my drawing, but essentially it has the same parts. And I'll just run through it just so that you can see where the different bits are. Here we've got the afferent arteriole going into the glomerulus, which is this bundle here, and then you've got the efferent arteriole coming out of it. This is the Bowman's capsule. It's the thing that surrounds, the glomerulus here. That's the Bowman's capsule. This is the tubule that's coming off it, so it's the nearest to the Bowman's capsule, so that would be the proximal convoluted tubule. The big drop is sort of easy to see. There you've got the loop of Henle. This is the tubule that comes off the loop of Henle, but it's furthest away from the Bowman's capsule, so that would be the distal convoluted tubule. And finally you've got the collecting ducts that go down, drawing green, down here towards the ureter and get rid of the waste products. The one thing that I didn't sort of show on the diagram, which I hope you can pick up on, is after the efferent arteriole leaves the glomerulus, if you track this, you can see that this arteriole and this capillary winds its way around the rest of the nephron, which I didn't actually include on my diagram, which is really, really important. And I'm trying to shade it in here to show you, because remember what I said about the basic principles of the nephron is that big things that are still left in the blood need to go back into the nephron. Waste products need to get put back into the nephron. But also, we've got a lot of stuff in this nephron here, this tube that we've filtered out, that need actually to get put back into the blood. We need to keep it, so we need to get that back into the blood. And that's one thing that I really needed to mention before we move on. So ultrafiltration, and I'll highlight it in purple, occurs, it says, at the renal corpuscle, which basically means the joint between the Bowman's capsule and the glomerulus, okay, which, let me show you, is more specifically right in there. That's where ultrafiltration happens. This bit is like the sieve on our diagram, where all the small things go through, but all the big things stay in the blood. That's where we talked about at the start. So molecules smaller than 3 nanometers in diameter, like water, glucose, and amino acids, small amino acids, can go into what's called the filtrate. And the filtrate is the product. So it's the stuff that comes down the tube. That's the filtrate. It's the stuff that makes its way down this snaky tube, down into loop of Henle, etc. That's what we call the filtrate. Larger molecules are barred from entering the PCT, like red blood cells, large proteins, hormones, that kind of thing, drugs. That stays in the blood, and that will go out into the efferent arteriole, and it will transport around this blood supply until it's pumped back into the filtrate that's going around this tube, so that it can be passed out to the ureter. That's the overview of the sort of term. So let's have a look at the structure and function. How does it do that? Well, the first thing that we should look at really is this structure of the glomerulus, because one of the first things that allows us to filter small things out of the blood to start with is high pressure. In the glomerulus and the Bowman's capsule, it goes under really, really high pressure, and one of the main reasons for this is the size of the afferent arteriole and the size of the efferent arteriole. And you can see that this going in is wide, this going out is narrow. And what that does is it builds up high pressure inside the glomerulus, because there's less chance of this escaping out of the efferent arteriole, so it builds up high pressure, and that's what causes, or that's one reason why the glomerulus and the Bowman's capsule is, specifically the glomerulus is suited to ultrafiltration. It forces the blood under high pressure, and that allows us to filter out the small molecules that are in the blood. Well, what else do we have? Well, the lining of the glomerulus, which is this, remember, is the capillaries inside here, and all tightened up in a ball, are fenestrated. A fancy word. What does fenestrated mean? Well, if you look at the diagram here, a fenestrated capillary has lots of holes in it, and that's what you'd expect to see in a sieve. If you're sieving some flour, for example, you're making pancakes in the morning, you want to make that flour nice and thin, it only lets the small stuff through. And you can see that the capillaries in the glomerulus have tiny holes and tiny pores in them, which only lets the small stuff through. So if you put stuff under high pressure and you have little small holes to let things through, that's going to really suit ultrafiltration. And when your blood rushes through there, all the small bits and bobs will filter in through. The big stuff will continue on through the efferent arteriole and back through the blood supply. The next structural feature refers to the Bowman's capsule. So this is the first time really they're talking about the nephron. This is, remember, the Bowman's capsule is the first part of the nephron. And one of the structural features of the Bowman's capsule is it contains cells called podocytes. Now, pod generally, relates to feet, and podocytes basically wrap around. Hopefully, you can see it in this diagram here. They wrap around that fenestrated capillary, and they have little filtration slits in them. Again, to stop big molecules coming through. Now, that's a super zoomed in version. You'd only see that. That's really, essentially it's microscopic and it's not letting big things through. So that's one of the other structural features of the Bowman's capsule that allows for ultrafiltration. So before I move on to selective reabsorption then, I'll summarise the structural features that allows ultrafiltration to happen. You've got the high pressure, you've got the fenestrated capillaries, and you've got the podocytes, which help also filter, and that's the three structural features that I'd expect you to write about in your question, and your answer, should I say. The second process that we will talk about for the test, for the question, is selective reabsorption. What that means in terms of our layman's term analogy is that we've got lots of useful stuff that have gone into the nephron, that's been filtered out. There's lots of small stuff like glucose that's now gone into our nephron that's actually headed to form our wee, and we don't want that, because glucose and amino acids and all these small molecules are really important for the body, so it would be stupid of us to get rid of it. So what we now have to do is selectively reabsorb that stuff back into the blood so that we can keep it, use it, and be happy. So let's have a look at how that works. So as I've said, selective reabsorption is the process by which we move substances selectively back in from the filtrate back into the blood. We want to keep the good stuff and not get rid of it. Now, it actually takes place along the proximal convoluted tubule, the distal convoluted tubule, and the collecting duct. So it takes place here, here, and all along here. So it's never really just a one-stop shop in terms of the kidney. And if you really look into it, it's extremely complicated. I need to sort of simplify it into a level 3 solid understanding. The main area for selective reabsorption occurs in the proximal convoluted tubule, so it's okay for us to focus just on that at this level. Okay, all of the glucose is reabsorbed. What do we need glucose for? For respiration. We've worked hard to get the food and we need energy, so why would we get rid of it in our wee? And obviously if there's glucose in the wee, which is often what people test for, then there may be a problem along the line, such as diabetes, for example. All amino acids, the cells need that to create their proteins. There's 20 essentially amino acids in the body. Why would we get rid of them? They create our receptors, they create our hormones, they create our cytoskeleton, they create neurotransmitters. It would be pointless to get rid of them. Again, water. 99% of water gets reabsorbed in the proximal convoluted tubule. It gets squashed through this fenestrated capillary, and then, oh, by the way, we need that, so we pump it back, or we let it back into the blood. Sodium and everything else becomes the urine. Okay, and I'll let you read that in to your own devices. So that is the term selective reabsorption. We keep the good stuff, we get rid of the bad stuff. So let's have a look at the structure and function. Walls of the proximal convoluted tubule, which is here, let's not forget, are formed by cubed epithelial cells on the outside. Okay, so you can see on the diagram here we've got 1, 2, 3, 4, 5 cuboidal cells, cube-shaped cells. Large number of mitochondria. Why might you think that would be present? Why would there be a large number of mitochondria? Think about that in your own head if you would. The answer, or there is a clue, in this diagram down here below? And the answer is active transport. We need it to move molecules. We've got protein pumps in there that are moving things from low concentrations to high concentrations, so we need to use active transport. There is a dense microvilli, so you can see these microvilli on the end. Why do we need microvilli? Well, you should be shouting at the screen right now. It's for increased surface area, and it is right there for reabsorbing water and solutes. You know, it's a common thing we've seen throughout the year that an increased surface area increases the rate of exchange of substances across that membrane. So it's really important, particularly when we're saying that the proximal convoluted tubule is the main site of reabsorption. So we need it. We need that surface area to be able to soak up all that goodness that's actually been passed into the tubule, and we need to get it back before it gets down the waste pipe, shall we say. Protein pumps use active transport to reabsorb sodium, so from low to high. Aquaporins, we've seen that again, allows reabsorption of water. We've got passive transport. We know that the phospholipids are diffusible, if you like, to small lipid soluble molecules. And this leaky gap junctions relates to spaces between cells so that actual molecules can fit between cells. So let's have a little bit more of a look at what happens. Now, don't be confused at this diagram. It looks confusing, but if we take each process in turn, we can see actually what's happening. Here down the left hand side is the tubule. I wrote it in blue, T for tubule. This is the inside of the proximal convoluted tubule. The middle section are the cells of the tubule itself. So this middle section, let me here see, these are the cells of the tubule, and this is the blood. You can see on the right hand side, the blood. So selective reabsorption basically means we're taking this small stuff or this good stuff and we need to put it back in the blood so that the body can keep it. That's what selective reabsorption is. So we've got a few examples here, and down the right hand side is the explanations. But hopefully you can spot water here going through these proteins here, and you will not be forgiven for assuming that they're aquaporins. And you can see over here for number 4, water's reabsorbed by aquaporins. What else have we got? We've got glucose, amino acids, some ions. Here you can see that it's sort of it's actually co-transport, and I'll leave you to find out what that is. It's not particularly difficult, but have a look at it. What is co-transport? Now you can see from the key at the bottom, it is a secondary type of active transport, and that should flag up in your brain that that's using ATP and it's using energy to do that. So have a look at it. Lipid soluble substances, that's the stuff that's going directly through the cell membrane. It's putting back into the blood. And interestingly enough, here's a tight junction. So that's the junction between one cell and another. You can see that actually stuff like chloride and calcium and potassium can squeeze in between the two cells, and that's called the paracellular route back into the bloodstream. So this is basically it. This is a summary of how stuff gets into or back into the blood. It's the good stuff that we need. We've shoved it under high pressure into a sort of collecting duct that's on its way to the garbage chute. Well, before we get there, we need to pull back all the good stuff that we need. Have a read down this right hand side and it should make sense, but hopefully I've done a decent job of explaining what's going on there. If you're explaining how the structure is suited to its function, you need to relate it to its structure. So you've got aquaporins, you've got gaps between the cells, you've got protein pumps that pump stuff in and out, you've got the cell membrane which is selectively permeable anyway. It's all stuff that you know, and I'd expect you to be writing about it. And that's just a summary of what I've just said on the last slide, so that you can use that for a bit of text if you want an official wording of stuff. And here is a nice summary of a table with different substances that are reabsorbed and the method by which they are reabsorbed. So pause the video, have a look, and that's for the proximal convoluted tubule. So the loop of Henle is really quite easy to distinguish. It's the one that looks like a roller coaster. It drops down and comes back out. So the proximal convoluted tubule, we're going to be going this way. So here's the PCT, drops down into the loop and comes out as the distal convoluted tubule. Now remember where we are in terms of the nephron. Anything sort of lower than this is the medulla. Anything above it is the cortex. So the nephron drops down into the medulla and out back into the cortex through the distal convoluted tubule. I'm going to simplify it really well for you, hopefully, so that it's a lot more complicated than I'm making it, but I'm just going to sort of simplify it because that's what's needed at this level. Now we've got two portions of the loop of Henle. We've got the descending portion and the ascending portion. That means going down and going up. We're going to take the ascending portion first, and the ascending portion has protein pumps on the membrane of the cells that make up this tubule. And what it does, it pumps salt into the medulla. And then what that does, it creates a salt bath. So the medulla is super salty, and I seem to remember reading that medulla is something like 5 times saltier than salt water. Seawater. So it's, you can imagine how salty that's going to be. Now the reason why that does that is because on the descending portion, the cells of the descending portion have lots of aquaporins in their membrane. Now if you were water travelling down the descending portion of the tubule, you've got through a reabsorption, you're still in the filtrate, you're coming down into this salty area. What's going to happen to this water? Well, you're not wrong in thinking that this is very dilute and this is very concentrated. So this is really salty in the medulla. So what that does is it draws the water out through these aquaporins into the medulla. All right, we want to keep this water. It's really, really important for us to survive. Our cells need the water to be able to survive. So by forming this salt bath, what you're doing is creating a concentration that's really high in the medulla, so that the water that travels down this descending loop wants to balance out the potential. And so what happens is it gets put back into the blood, and we retain this water to be delivered around the body. Okay. Now when we talk about structure and function, the ascending loop has protein pumps on it which pumps out salt. It's not permeable to water, so water is not able to leave this portion. So once the water gets round here, it's not able to leave. There are no aquaporins. Okay, but the fact is that this water escapes in the descending portion, and so by the time it comes down here, we've got really concentrated filtrate. Okay, because all the water's been really sort of taken out of it, and that is pretty much the function of the loop of Henle, with the extra part that the descending loop is really permeable to water because it has lots of aquaporins on the walls that make up the tubule. So this working together enables us to retain water really well. We create a salt bath to create the potential difference, and we have aquaporins on this membrane so that water's able to escape and we can retain the water. I've hopefully done a decent job, and hopefully you understand what's going on in the loop of Henle. So finally, after the molecules have been filtered, they've been either reabsorbed and they've gone through the loop of Henle and they've come up through that ascending loop, they're finally ready to be excreted. So what we're going to look at is how things get put back into the filtrate, So if you remember, we'll focus on the distal convoluted tubule. Here's the distal convoluted tubule and here's the collecting duct. We've got stuff coming down here that's ready for excretion, but we've also got stuff in the blood that's, remember, the big stuff that didn't get filtered, that we've got to actually put back into the filter so that then it can go down the collecting duct. So we've got to look at this last process of how actually things do get excreted. And it's relatively quite straightforward and less interesting, I would say, than the other two sort of processes, but essentially mainly they're actively pumped, so they move from a low concentration to a high concentration. Protein pumps basically pump them across the membrane into the filtrate so that we can get rid of them, in particular drugs, because that's, I suppose, stuff that we've been talking about in infectious disease, which is important. So it actually occurs along all sections really of the nephron, but we'll focus on this because I think it helps to separate the different sections. And he said that's it. It basically, all the stuff from the blood gets pumped back into the filtrate, which then goes down the collecting duct to the ureter. So we need to put that in some kind of official language, academic language, for your answer. One of the things that I need to mention is that the excretion of water is under the banner of homeostasis. And remember, that's balance. It's a balance of water. So if we're dehydrated, we need to retain water, we need to keep the water inside our body, and therefore we will go to the toilet less frequently. If you start drinking lots and lots of water, then your body will go, we're drinking too much water, so we need to get rid of it. And so that's why it comes under homeostasis. So let's have a little bit of a look at that. So it forms definitely the process of SREA, so stimulus, receptor, integrator, effector, and response. So the stimulus would be, in this case, that let's say that we're thirsty, we're dehydrated, so our water potential, our water content goes down. That would be the stimulus, our water levels go down. In the hypothalamus, osmoreceptors, osmo obviously water, and receptors meaning a receptor, detects that and does something about it. What it does, it tells the posterior pituitary gland. Now the posterior pituitary gland is the one behind it. If you look at this little thing here, you'll see that that's the pituitary gland. The posterior one is the one at the back. I often call it the ball sack of the brain because it looks like a ball sack. That's the anterior pituitary gland. This is the posterior, and that's the one that releases a hormone called ADH, antidiuretic hormone. ADH causes the collecting ducts to move aquaporins from the interior of the cells to the external surface where these proteins are inserted to their cell membrane. So if you've got a load of cells on this collecting duct here, okay, and remember, we're thirsty. We need to keep the water inside our bodies, right? So this hormone triggers these cells to produce aquaporins on their cell membranes. So as any water that comes down here to the collecting duct, remember that's going to form urine in the bladder, this water then is allowed to, through osmosis, to be put back into the blood, because the hormones triggered these aquaporins to be present on the cell membrane. Okay, so that enables us to retain water. Now don't forget that this loop of Henle is in the medulla, and so this is extremely salty, and that aids the osmosis of water back into the blood, and that's why we would retain the blood. So what you would see is if we were dehydrated, We wouldn't be going to the toilet as much, there wouldn't be as much urine produced, and we would definitely see a much greener, darker urine than what we might see in terms of a clear, yellowy substance, if that makes sense, because all of the water comes out and it makes it more concentrated in terms of the darker green. Okay, so let's just go through that again. Stimulus, we are dehydrated. Receptor, osmoreceptors in the hypothalamus cause the integrator, i.e. the posterior pituitary gland, to release antidiuretic hormone, which is the effector. The response is that aquaporins are placed on the cell membrane of the collecting duct, which, as water travels down the collecting duct, means that water is recycled back into the body so that we are not losing excess water, and therefore has the response of increasing our hydration levels back to the set point. Now, as is always the case in homeostasis, let's say that we are completely overhydrated, and it would work the opposite way round, and I'm hopefully not offending your intelligence by going through this. If we are overhydrated, so our hydration levels go up too much, osmoreceptors detect that. They tell the hypothalamus, the hypothalamus tells the posterior pituitary gland not to release diuretic hormone, antidiuretic hormone. That means the cells of the collecting duct will not produce aquaporins, and therefore the water will travel down the collecting duct into the bladder, and therefore that will reduce our hydration back to the set point, because we'll have lots of clear wee and getting rid of our water. And that is the homeostasis of water, essentially. That's how we're able to maintain the water levels within our body.